Welcome back to my talks. And today, third lecture of chapter 6, the search for genetic material. Last two we did about DNA, the structure of DNA, the nucleophile structure, and now we'll do the search for genetic material. Okay, now here till now it was not confirmed whether the DNA was the genetic material. Okay, so the scientists were in a search, okay, to find out whether it was the DNA, the genetic material, or the proteins, because they knew that something was present in the cell. For Mendel's experiment also, it was found that yes, hereditary changes did take place, but what is it present in the cell which brings about this? Okay, is it the proteins, the DNA, RNA? What is it which causes change? Okay, so that search was going on, and Fred Griffith is the scientist. Fred Griffith in 1928 performed experiment with Streptococcus pneumonia. Okay, Streptococcus pneumoniae is the one which causes bacteria, which causes pneumonia. Okay, so he performed quite a lot of experiments based upon this Streptococcus pneumonia. Okay, and studying his experiments and concluding his experiments, we will come to know actually that what is the genetic material. So over here we are going to study that. Okay, so what did he do? He cultured, okay, artificial culturing, no, on the culture plate he tried to grow Streptococcus pneumonia. Okay, so when he grew Streptococcus, normally the Streptococcus pneumonia, okay, they are of two types. This pneumonia, it is of two types. So you can say these bacteria, Streptococcus bacteria are of two types. Okay, one is of S type, that is smooth wall type. Okay, and this smooth wall, they have a polysaccharide coat. They have a coating which makes it very slippery. So this is smooth wall and this is rough wall. Okay, this does not have any coating. So these are of two types. So finally when he cultured, he found that the cultured plate had two types of Streptococcus bacteria. Okay, there were few with colonies of smooth walled and a colonies of rough walled. Okay, so what did he do? He first took smooth walled. Okay, smooth walled he took, and he took a rat. Okay, a mice, a rat. Rat is there, mice is there in your book. I am telling you rat. Okay, so he took a rat, and this S, he took it into a syringe. Okay, जिसे injection देते ना, the syringe. He took this S वाला smooth walled. This is smooth walled bacteria. Okay, so smooth walled, we are going to write S. So that is why I am putting S. So smooth walled bacteria, and then he infuses into the rat, and he sees that the rat dies. Okay? So finally then after that he takes only R and he puts it into the rat. Okay, again in a different syringe, and the rat is alive. He does not die. Okay, after that what does he do? We know that when something we boil or we heat, it gets killed. So what does he do? He takes S, heat killed. Okay, heat killed, he kills it. Okay, and then he takes it into a syringe. Okay, and then again he takes a rat and he tries to inject it. So after killing it, after killing that bacteria, again that bacteria he takes and puts it into the rat. Okay, and the rat is alive. Got it? So now he may conclude that this has become non-virulent. Okay, this one is virulent, this one is virulent. Okay, and this R is non-virulent. Non-virulent, because this does not cause death, and this one is also become non-virulent now. I hope up till here it is clear to you, children. Okay, after that what he does, he takes this S hit killed and he takes R, because both does not kill. I'll use a different pen. With both they are alive. They are alive. So what does he do? He takes this R and he takes S heat killed. I'll just write it over here with reading. You can know. S, okay, heat killed, and he takes plus he takes R. Okay, and then he injects into the rat. Injects into the rat, and he sees that the rat dies. Got it, children? This is the third story. So he takes this heat killed smooth walled bacteria, and he takes this R rough walled bacteria, injects into the rat, and he sees that the rat dies. Now he concludes there is something into that, there is something into, something is wrong, something has happened, some transformation has taken place that has brought about the death of this particular rat. So he tries to find out, and he sees that when the DNA from it is found out, actually what is the reason of the death of the rat, it is found that the bacteria are there, and the bacteria is smooth-walled. Got it? The bacteria which was found from the rat's body was found smooth-walled. It means it has developed that smooth-walled coating, that mucus coating it has got. Okay, so now what is it which is responsible for that? So he also said that maybe something from this R, okay, or something from this S heat killed, something was still alive in this smooth walled heat killed, okay, which transformed this R, the rough walled, okay, and developed a coat around it. Are you getting it, children? Okay, something which was still present in the smooth-walled heat-killed bacteria, which transformed, which brought about a transformation into this rough-walled, okay, which was non-virulent, and developed a smooth wall around it and made it virulent. Okay, so that the rat got killed. Okay, so now he wanted to find out what is it which had transformed this rough-walled these bacteria. Okay, what was it? Is it protein, DNA, RNA? What was it? Up till here was the Fred Griffith's experiment. So this we finished. The second one is that there are some scientists who wanted to find out, along with this Fred Griffith's experiment, that what is it which is responsible for the transforming principle, for the transformation of the rough walled into smooth walled. Okay, so is it the protein, is it DNA, RNA, what is it which is responsible? So finally they took the different, you can say the enzymes, okay, and proteases. With the proteases they tried to find out whether protein was responsible, okay, for this transformation, and they got a negative result. With the RNAs they found out that was RNA responsible? No. And finally it was the DNA actually which was responsible. It means the DNA brought about the change into this smooth walled, into this rough walled. Okay, the smooth heat-killed bacteria transformed the rough walled and made it smooth. So it was DNA. So the search for the genetic material, okay, more about came to an end over here. Okay, so these 2 points, again let me refer to your book. Okay, the search for a genetic material and biochemical characteristics of transforming principle, we do it over here. Okay, and there is another experiment, Hershey and Chase experiment. The genetic material is DNA. There are certainly 2 experiments. Okay, this experiment is also very important, and Hershey and Chase experiment is also very important. So this the search of genetic material, and the other it is confirmed that the genetic material is DNA. That we are going to do in the next class. Up till here I wind up the class, okay, as the search material for genetic, the search for genetic material, okay, and we confirm that it is the DNA which is the genetic material, okay, which brings about the transformation. So thank you. You must have enjoyed the class. Keep watching. Thank you so much.